The remainder of this lecture is going to be basically unique to eukaryotes. And we're going to talk about what happens to the messenger RNA after it's done in transcription. As I said a minute ago, in bacteria, they can actually transcription and translation can actually happen at exactly the same time. So this is, it's actually the very last slide. It's the very last slide in the lecture. So on the bottom is actually the DNA, and then this string is the messenger RNA. And as you can see, the messenger RNA hasn't even dissociated from the DNA yet, and we've got ribosomes on it for translation. So in bacteria, you basically have that situation of one gene, one protein. So as soon as that messenger RNA for the protein is being transcribed, then ribosomes can come in and translate it. And again, this is partly because the bacterial chromosomes are not isolated in a nucleus, and they can do this. So you'll also notice that there are, in the same piece of DNA, multiple transcripts. So usually in a cell, as long as the gene is turned on, messenger RNA is going to continue to be produced. And that's what's happening here. We're getting multiple mRNAs from the same strand. But eukaryotes, because of the nucleus, are a little bit different. So once termination happens and our messenger RNA comes off, then we have to do what we call RNA processing. So we have to do some things to that mRNA molecule to get it ready for translation. So there's a couple of things that happens in eukaryotes that are basically modifications of that original messenger RNA. Some of those modifications are related to actually moving the messenger RNA from the nucleus out to the ribosome. And then some of them are sort of processing of the gene itself. So the first thing we're going to do is actually make sure that our messenger RNA is fully ready to be transcribed. In a eukaryotic gene, there are two types of sequences once we get into that RNA coding region. So there are two types. We've got exons and we've got introns. And the function of these, the main function is that in a eukaryotic cell, you can have different combinations of these coding regions put together to make different genes, or sorry, different proteins. So what we get in eukaryotes is that a single gene can actually potentially code for multiple different proteins. So down here on the bottom, we've got a gene. There's two different examples here, but there are these gray sequences that indicate an intron, and then the darker blue-gray that indicate the exon. So what's going to happen here is these sequence regions called introns are going to be removed, and the exons are going to be put together. So as we're processing this mRNA, we're going to be taking out those introns, and then we're also going to be doing some other modifications. So introns and exons are going to both be transcribed. They're part of that original transcription unit, but then the introns are going to be removed. In eukaryotic genes, introns are really common. You don't tend to find them in bacterial or prokaryotic cells. Some of these introns are sort of structural. They're there for structural reasons, like maybe they're important in like DNA packing, and so they need to be there to help the chromosome pack correctly at the right place. And then we'll talk about alternating in just a minute. So I feel like this should be at the beginning, but our textbook plops it right in the middle, and that is what it means to actually be a gene. So throughout our textbook, we talk basically, without knowing it, you have found several different definitions of a gene. So chapter 3, when we talked about Gregor Mendel and genotypes and phenotypes, we were defining a gene as basically a unit of inheritance. It was a particular sequence that codes for a characteristic, and that sequence gets passed on to our offspring. In chapter 8, we talked about, we didn't define it very well, but we talked about how a gene is a set of nucleotides. So there's a specific set or sequence of nucleotides that is going to encode that characteristic or inherited factor that we talked about in chapter 3. Then this chapter and the next chapter, we're talking about a gene as a set of nucleotides that's going to allow us to produce an amino acid chain and then a protein. So we're kind of evolving what we mean by genes depending on how we are studying them. So are we studying them for inheritance, or are we studying them from a molecular biology standpoint? So in 1902, a man named Archibald Garrod hypothesized that genes are somehow being used to make proteins. And so I mentioned at the beginning the one gene, one protein hypothesis. That was kind of what he was saying, that whatever the genetic material is, because we didn't know quite yet in 1902, but whatever that genetic material is likely coding for a particular protein. But then I mentioned that now we recognize that a single sequence can encode for multiple proteins. And introns just kind of complicate that because introns are not coding for amino acids.
So what exactly is introns?
We'll talk about them in just a second. Okay, so from a molecular biology standpoint, we are going to define a gene or basically define a gene by its characteristics. So a gene is going to be a particular sequence that needs to be replicated and passed on to the next generation. A gene also has to have potential for variation. So this is where our nucleotide sequence comes in, and then also our nucleotides coding for our amino acid. And then sort of related to that, and specific for this chapter, a gene is going to be used to produce a particular phenotype. One of the handouts that I gave you, table 10.4, this is a summary of the different types of modifications that we're going to go through. So it gives just a really brief description of all of the modifications that we need to do for the messenger RNA. Okay, so we talked about our transcription unit, the promoter, followed by our coding sequence, and then our terminator. All of those get transcribed, but not all of them get translated. So we had our promoter. So if we draw our transcription unit, we had our promoter, we had our RNA transcript unit, and then we have our terminator. And then this is what we would call, or sorry, we're going to produce a messenger RNA transcript. So from there, we get our messenger RNA. And then we have the promoter and the terminator are going to be transcribed. But it is not, I'm sorry, it is only that center, that RNA transcript unit that is actually going to be translated. So our mRNA is going to have our promoter and our terminator. And it is also going to have introns and exons. So what actually gets translated are just the exons. All right, so in our initial messenger RNA transcript, we have the promoter, we have the terminator. Those sequences are going to be removed. And in their place, we are going to add something called a cap and a tail, and we'll talk about those in a minute. We're also going to remove the introns. So the introns are non-coding regions. So they are not going to be part of our final protein transcript. Non-coding regions that are removed from the final mRNA. Our exons are going to become part of the final messenger RNA that's actually going to be transcribed. So the names come from the name exon comes from expressed sequence, meaning that's the sequence that we're going to use to actually produce our protein and our phenotype. The word intron comes from the name intervening or in between sequence. In other words, it's the sequence that's between the exons that are not part of the final coding DNA.
Could there be mutations? Yes. Like if a intron was.
So if there's a mutation in an intron, it's not going to affect the final protein. Because it's not part of the mRNA that's going to get transcribed. translated, sorry. It's not part of the mRNA that's gonna get translated. However, if there is a mutation in an exon, then yes, that could affect the final protein. Is that what you were asking? Okay. So, our initial messenger RNA that comes off is called a pre-RNA. And then the final one, the one that's going to go to the ribosome, is called a mature RNA. So here we've got our gene sequence with our promoter, our introns and our exons, and then our terminator sequence at the end. And then we've got the pre-mRNA. So it's got a little bit of the promoter sequence there. And then it's got our introns and our exons. And then we're going to process it. We're going to do a couple of things to get it all ready. And then the final product is going to be our mature mRNA. So this diagram kind of goes along with the handout that I gave you that talks about the different post-transcription modifications. So this kind of steps through. So we've got our original one, we've got our transcript. Here we're adding a cap. Here we're adding a tail. And then here we are removing our introns. All right, so we're going to take this pre-mRNA, and the first thing we are going to do is add a 5' guanine cap. So that's the first thing we're going to do. So step C there, we're going to add this cap. So, oh. We don't need that right now. I'm not sure what's going on there. Okay. So the first thing we're going to do is add in this cap. We call it a guanine cap because it is a special modified guanine molecule. So here is our guanine. So just a regular guanine molecule as a base. We've got the ribose, the nitrogenous base, and then in place of the single phosphate that we would use if we were building a DNA or an RNA, we have this special group that's attached to it. So this is added to that 5' end of our RNA molecule. So there's a couple of reasons for this cap. One is that it is important for the ribosome to recognize the start of the mRNA. So during translation initiation, the ribosome is going to recognize the 5' cap. So it's actually going to be important in bringing all of the pieces of the ribosome together. And then it also helps increase the stability of the messenger RNA. So in our cells are constantly producing messenger RNA. And as soon as the cell is done with them, the cell's going to degrade them using a nuclease. And the components of the messenger RNA are going to be recycled into new RNA molecules. So in order to make sure that the brand new messenger RNAs are not degraded, part of the function of the 5' cap is basically to protect that end from degradation. And the next thing we're going to do, adding a tail, is also going to do the same thing. It's going to protect that messenger RNA so it doesn't get degraded as soon as it's produced. All right, so once we get the cap on, we're going to the other end, we are going to add what we call a poly A tail. So poly A means many adenines. So we are going to add a whole bunch of adenines to the end. This can be anywhere from 50 to 250 nucleotides to the 3' end of the messenger RNA. So just like the cap, it's there for stability and also to make sure that that messenger RNA is not broken down when it's not ready to be broken down in the cell yet. So this image shows just this string of adenines attached to the end. So the last thing we're going to do is the exon splicing. So what we're going to do in this step is remove those non-coding introns and link the exons together. So in order to do that, the cell has to recognize the point between the intron and the exon, where the end of the intron starts, where the beginning of the exon starts. Just like those consensus sequences at the beginning of our molecule at the promoter region, there are also consensus sequences that happen between the introns and the exons. So these non-coding sequences are going to be identified by the presence of these consensus sequences, and they're going to be recognized by something called a spliceosome. So a spliceosome is a big enzyme composed of at least 300 proteins, so really, really big enzyme. And its job is to recognize those non-coding regions and pull out those regions and then put together the exon. So there's a whole bunch of steps here. It has to bind, it has to cut, has to hold the pieces together until it can stick them end to end. So it's big and it's bulky, and its job is to identify the proper combination of those introns and exons. Our textbook does not have an image of this. I've never seen like an actual like sort of like computer digital image of this. I've seen kind of like cartoon diagrams of this. Our book doesn't even bother. But basically what it's going to do is recognize this consensus sequence, cut at a particular spot. Let that intron fall out and then pull the exons together. All right. So 2 steps here. We're going to cut the 5' end of that non-coding intron and then cut the 3' end. What's going to happen there is that intron is basically going to kind of become unstable. Our textbook goes into this process of the intron like folding back on itself. And the purpose of that is so that the cell knows that it can degrade that intron, that intron's been pulled out, it's no longer needed, and it can be broken down into its components. And then that molecule is going to take and join, kind of like DNA ligase, join the 2 fragments, the 2 exons, pull them together. So those introns, I like to say introns means it's staying in the nucleus. So it's going to stay in the nucleus, get broken down there, whereas the exons are going to exit the nucleus. They're going to be spliced together and leave the nucleus to go be transcribed. Did I answer your question about what an intron is? It's sort of, yeah. So it's basically just a non-coding sequence. And the purpose of them, one of the purposes is something called alternative splicing. Before I get to what alternative splicing is, in some genes, there are regions that are there simply because for another reason. They're there because they are part of like binding of DNA polymerase, or they're there because there's structural reason within the chromosome that they have to be there. So they're there outside of being there for the actual gene itself. However, sometimes, depending on the gene, sometimes a region will be an intron and sometimes it will be an exon. So the main reason for introns is something called alternative splicing or alternative processing. So alternative splicing says that you can link together your exons in different combinations. So what that means is that you can have a single gene in your DNA that codes for multiple proteins by linking together different exons. So what is an exon for one protein of the same sequence might be an intron for another protein coming from the same sequence. So it's not always that the intron doesn't do anything. It may be that the intron is simply not being used for that particular protein. So if we go back and look over here, in this gene, we have exons 1, 2, and 3 for one particular protein. But for a different protein, we might link together exon 1, exon 2, and then intron. So for a single gene, you might have different introns and exons. So this image down at the bottom just kind of is illustrating the different ways that you could link together your genes. So it's estimated that 60% of eukaryotic proteins are alternate splicing products. So for example, down here, we've got basically exons 1, 2, and 3. We could link them all together. Or we could leave out this sequence and just link 1 and 2. Or we could leave in a different intron and get a completely different protein product. So it's not always that those introns don't code for anything. It's just they're not coding for the particular protein version that's being produced right now. we are not going to get too far into transcription, but I'm at least going to introduce it. Oh, did you catch me? You did. You did. You caught me.
I was just confused. So I was like, did we not just do transcription?
Oh, Lindsay caught me. Okay. All right, translation. We're going to introduce translation. We won't get too far, but before I do that, are there any questions on transcription or mRNA processing? So think on those introns, think on alternate splicing, and if you have questions on Wednesday, we can definitely come back to this. And I actually found an article that I'm going to assign for extra credit whenever I get my life together and write it that is about alternative splicing. Okay. All right. So we talked about producing messenger RNA, but there are other types of RNA that we can get out of transcription. And one of them is transfer RNA. So transfer RNA is going to be required in the process of translation, and its job is going to be sort of the translator between the messenger RNA sequence, what is coded in the messenger RNA, and what string of amino acids is actually put together in our protein sequence. So just like all RNAs, tRNA is single-stranded. So if you start at one end and follow it through, start at one end, follow it through, you'll see it's got some loops and turns, but it is all just one single length of nucleic acid. So sometimes we'll see it like this. Your textbook likes to simplify it and make this like blob shape. This is sort of the simplified double helix structure. You'll notice it does kind of twist. It's still a nucleotide, so it's still, when it is double-stranded, it will twist like DNA does. But depending on how far the complement area goes, depends on how much it would actually twist. This is if you took the molecule and laid it flat. So a couple of key features of transfer RNA. On one end, we have the amino acid attachment site. So the job of transfer RNA is to bring in the amino acids that we need to actually build our protein. So those amino acids are going to be attached on this end. On the other end, we have what we call an anticodon. So the anticodon is complementary to the messenger RNA. So the way that the transfer RNA is working is that the transfer RNA is essentially reading the messenger RNA sequence and bringing in the appropriate amino acid. So we've got several complements happening. We've got DNA producing the complement of DNA, which is the mRNA, and then the anticodon, which is complementary to the messenger RNA. This is one of the handouts that I gave you with the transcription translation packet. So if you look at this, the first thing you're going to do is make the DNA complement to make your double-stranded DNA. Then you're going to make a messenger RNA. Then you're going to make a transfer RNA. And they're all just complements of one another. And then you're going to use your messenger RNA code. Your messenger RNA code is the one that's going to code for the protein. Yes.
So is the difference between the anticodon and the DNA like the T and U? Because if it's complementary to mRNA.
Yes, it gets confused.
Okay.
But don't think of it as in relation to the DNA because the tRNA actually never has anything to do.
Okay.
But yeah, it is technically the same as one of the DNA strands. All right. We've got one more slide in this chapter. So the other type of RNA that I mentioned very, very early on is ribosomal RNA. And ribosomal RNA is part of the ribosome. So the ribosome is made up of RNA molecules bound to protein. And there are two parts to the ribosome. There's the large subunit, which is this top part, and then there's the small subunit on the bottom. The ribosomal RNAs are another example of a highly conserved sequence. And there are a couple of types of eukaryotic RNA molecules, but in general, they're all really, really similar. So the function of the small subunit is going to be to bind the messenger RNA, and then the large subunit is where we're actually going to add in our amino acids. So on Wednesday, before we go through our lab, I'm going to lecture on transcription. It shouldn't actually take too long. And then as you work through your lab, we'll look at what happens to the actual sequences. So there's kind of two parts. There's the sequences where we've got all the complements happening. And then there's also the actual process that's going on to build our sequence. So we will look at that part of it on Wednesday as well. Did I have what? Another part you just found out.